My name is Rong, and from Case Western Reserve University from Cleveland, Ohio. So I represent this work on behalf of my PhD student, Yang Chen, who did most of the work and cannot come here because of visa problems. So this is the overview of our study. So in this study, we developed an integrated system for both disease gene prediction and drug discovery. So for the first component, we developed a network-based approach to integrate high-level disease phenotype information from multiple data resources. And then we combined the high-level phenotype information with known disease genetics and the functional protein interaction. The input for the first step is a disease, output is a ranked list of genes. Then we take the ranked list of genes for each disease, we link those genes with known drug target, and then rank drug for each diseases. So probably it's well known that disease genetics hold great potential for drug discovery and drug repositioning. Recent studies have shown that the rare Mendelian disease genetics is a rich resources for drug discovery, not only for monogenetic rare diseases, but also for common complex diseases. For example, recent studies show that for 131 drug targets that link to genetic diseases, 45, it's about 40% of them can find direct matches between its associated diseases and its drug indications. For the mismatched ones, those ones can be a source for drug repositioning or drug side effect prediction. The same is true for common complex disease genetics generated by genome-wide association studies. Very recent study showed that for 991 gene associated with disease, as shown by GWAS study, 781, about 80% of them are druggable. Among those 991 genes, 155 of them are associated with known drugs. Among these 151 genes associated drugs, like 63 of them can find a direct match between the associated genes, associated disease, and the drug indications. For the ones, mismatch can be a source for drug repositioning. Since a lot of genetic mechanism of many diseases still remain partially or fully unknown, I argue that computational predicted disease genetics in combination with the disease genetics from what we know hold great potential for drug discovery. We demonstrate this point in one of our recent studies. In this study, we developed a novel algorithm by modeling the host and the parasite genetic interaction, and then we predict the genes for malaria. And then from the top predicted genes, we found it contains are highly enriched for known drug targets in general, and also enriched for malaria drug target in specific. So why this study is very specific for infectious disease like malaria that involve host and parasite interaction? In this study, we want to develop a very generalized disease prediction algorithm for many diseases. So our underlying hypothesis use disease phenotype to predict disease gene is that many studies shows that the high-level phenotype overlaps often reflects the underlying common genetic overlap among diseases. But the problem is using disease phenotype is that this kind of information is often scattered around in different data resources in different formats. For example, can be in OMIN text description of diseases, in medical ontology, in electronic medical records, in biomedical literature. So the challenge of in using high-level disease phenotype information is to extract those information from different resources and integrate them together. So computational disease genetic prediction is a highly active research area. So the method can be roughly divided into two categories. The first one is predict disease genes based on genetics and genomics, for example, protein-protein interaction, gene ontology, biopathways. The second category is use high-level disease phenotype in combination with disease genetics and genomics for prediction. So our approach is in the second category. We use disease phenotype. So our approach is different from the phenotype-driven approach is why this traditional approach often use one phenotype, use phenotype information from one resource. We use phenotype information from multiple data sources. So the key for our approach is a disease manifestation phenotype network that we recently constructed. And then we integrate, it's called DMN. Then we integrate this disease phenotype network with a widely used phenotype network called MIMI-MILER. MIMI-MILER was constructed from OMIN databases using text mining and techniques. So we combine these two disease networks together, and also we integrate with protein-protein interaction data, and the links. We also the link between this high-level disease phenotype network and the PPI network is what is known disease genetics from the OMIN databases. So I'll just briefly describe the disease manifestation network that we recently constructed. So we extracted about 50,000 disease manifestation pairs from the United Medical Language System Semantic Network. It's also called UMLs. UMLs, it consists of more than 160 biomedical ontologies. So for each disease, we create a feature vector consists of manifestation. For example, disease can be associated with like a fever or hypertension. And then for each pair of disease, we calculate the similarity for each pair of disease, just use cosine similarity. So then in the end, the disease DMN consists of 2,312 disease nodes and about 400,000 of edges. So we demonstrate that the disease that share high-level manifestation similarity also tend to share high genetics, share high genetics. And because we want to combine two existing phenotype networks, so we want to show if the knowledge captured in DMN is complementary to the knowledge in the MIMI-MILER. So we show it's largely complementary. So we just extend the standard network-based ranking algorithm to this heterogeneous network consists of 3 networks, two disease network and 1 protein-protein interaction network. So I'm not going to go to detail this algorithm. It's in the paper. So we perform the standard leave-out cross-validation and also de novo cross-validation. And so during the leave-one out cross, during the validation, so leave-one out cross-validation is remove one disease gene association each time, then to see if we can predict them back. So we be careful because the same disease can appear in both disease networks, so we want to make sure when we remove both edges to link them to disease network to the gene network. So this is de novo gene prediction. We just remove all the edges to see if we can predict them back. And so we then investigate if the phenome-driven approach, because we don't believe we can have a generalized approach that can work best for all the diseases. Then we want to see if whether like which disease approach work best. So we classify the diseases into 9 broad categories using ICD-10 codes. Then we repeat the experiment. So after we did like cross-validation and using known disease genetics to evaluate the algorithm, we also did case studies to see if our approach can find novel genes for diseases and find novel drug target for diseases. So we did this for Crohn's disease. So the results shown in the table. So our approach combined 2 disease phenotype together have much higher successful ratio than the one used one disease phenotype network. The same is true when we evaluate use AUC curves. And then we found the same trend for when we do de novo prediction. So basically, 2 networks performs better than using 1 network. Then we found this, the performance is really varies across disease categories. So the take-home message is whether or not you use phenotype approach for disease gene prediction, how much weight you want to put on disease phenotype is really depends on the disease you're studying. For example, the AUC curve is the best is for congenital malformation and the deformations. So that makes sense. And the lowest one is for cancer. That also makes sense because cancer phenotype really don't give you much insight about what's the underlying genetic causes. So the same is true for when we did this for the novel validation. So for Crohn disease, we evaluated our algorithm, whether or how our algorithm performs in terms of finding novel disease genes. Before, we just did a cross-validation use known disease genes from the same database like OMIN. Then we performed, if our algorithm can find the novel genes, we just use a independent, not independent, from another data source like a GWAS study. So we didn't use any data from GWAS study for our gene prediction, and then we use GWAS Crohn disease associated gene for novel prediction evaluation. Then we found that the top-ranked genes are enriched for genes from GWAS study. That integrated our algorithm may be able to find new genes for diseases. And also, we found the top genes are enriched for drug targets in general, and also top genes are enriched for FDA-approved drugs. Then we just manually look at the top-ranked drug candidates for Crohn's disease, and then we found a list of drugs currently are treating other disease, and these drugs maybe have potential for treat Crohn's disease. We did this by manual search literature for for this. So in summary, so in this study, we developed a Phenome-driven approach by combining high-level disease phenotype from multiple data resources with lower layer level genetic and genomics information. We demonstrate that the predicted genes have potential leading to new drug discovery. So in this study, we only integrate 2 disease phenotypes, but there are many other disease phenotypes. For example, disease phenotype from literature, from electronic medical records. So in the future, we're going to want to integrate additional disease phenotypes to further improve our networks. And also from this study, because we found that the performance is really varies across disease phenotypes, I think probably it's important to build a disease-specific prediction model. For example, you want to assign different weight. You have 3 networks. You want to transition to among different networks. You maybe assign different weight to each network for different diseases. So in the end, we predict a lot of new disease genetics. We want to build comprehensive models, combine our newly predicted disease genetics with known disease genetics, for example, from OMIN database, from GWAS studies, and then we want to integrate this disease-related genetics with drug-related data, and then build a comprehensive model for drug discovery. So this work, I would like, this work was mainly done by my PhD student, Yang Chen, and I also like thank for the funding resource for this work. Thanks for your attention.
So again, we're a little bit behind, but we can take a quick question while the next speaker gets set up.
Thank you. Did you see the overlapping between the disease terms and the manifestation of disease when you look into your HAS manifestations?
Yes. Actually, manifestation and the diseases, you cannot, it's all medical condition terms. And yeah, it's all about diseases, all about the medical condition.
So how would you take care of that when you see that a disease is associated to another manifestation, which is also a disease?
Yeah, that's a disease, but from the UMass network, like hypertension is a disease, and also is a manifestation for a lot of diseases. So in the UMass network, and they have disease manifestation association, say, given disease, what kind of manifestation associated with those disease?
And then you link that to the genes, and won't you end up with the genes that loop back to the same network?
I link the disease to the genes through OMIN databases.
Thank you.
Okay, thanks.